The overall goal of this procedure is to demonstrate the use of polymeric nanoparticles for the overexpression of therapeutic genes in stem cells using a murine hindlimb ischemia model. This is accomplished by first synthesizing biodegradable polymers through a two-step Michael addition reaction, followed by fabricating polymeric nanoparticles encoding therapeutic genes. The second step is to transfect human adipose-derived stem cells in vitro to overexpress vascular endothelial growth factor. Next, programmed stem cells are injected intramuscularly into an ischemic hindlimb for in situ production of therapeutic factors. The final step is to monitor cell survival and blood reperfusion in the ischemic limb over time using bioluminescence imaging and laser Doppler imaging. Ultimately, quantitative gene expression and histology can be performed to show localized expression of therapeutic factors and efficacy of tissue regeneration.
The advantage of this technique over conventional methods for angiogenesis is the use of stem cells as vehicles for drug delivery. This strategy allows us to employ the natural homing capacity of stem cells to migrate towards ischemia and also allows dynamic responses to microenvironmental cues.
The nanoparticles used for plasmid DNA delivery are highly efficient and biodegradable, affording for lower cytotoxicity and a larger number of intracellularly delivered DNA copies.
This technique can be applied to many clinically relevant cell-based therapeutics because it utilizes a biodegradable, non-viral vector to insert genes into autologous cells.